Okay, everyone, it's already 8 o'clock past 3 minutes in our time. So we have to start now. First of all, I would like to say good morning, everybody. Assalamualaikum warahmatullahi wabarakatuh. For the honorable, our special person, our special teachers today, Professor D. Unglap Silverthorn. Welcome, Professor. So, hello. So, we would like to say thank you very much for you, for your willingness to be here to give the lecture for our students and our faculty members, especially of physiology faculty members. So, Professor D, before you start the lecture, I would like to tell you that this lectures is part of our program. We call it the Insightful session with world-class professor. Actually, that we already invite 5 professors before you. That Professor Cheng Swimming, that I believe you already know here, know him, and Professor Noriyuki Koibuchi, and of course your best friend, Professor Robert G. Carroll.
Yes.
On last Thursday, he gave the lecture to us. And then also 2 professors of Indonesia, Professor Putu Gede Adiatmika and Professor Nengtina Kartina. And as you may know that our lecture today are broadcasting to YouTube. So, and we are inviting all of the faculty member of physiology from Indonesia. So maybe Dr. Adrianta are seeing you now, and Dr. Ermita and everyone of physiology teacher. And also this lecture also watching by the or following our home faculty member from Hardy, Dr. Harry, and anything else. So students, before Professor D start her lecture, so I would like to uh, introducing her to you. That kenapa tidak mau ya? Okay, this one, this is Professor D. Unglap Silverthorn, PhD. That you most of Indonesian medical student know Professor D from her book, a very famous book, Human Physiology of Integrated Approach. And Professor D is the professor of physiology from Dell Medical School, University of Texas, Austin, United States of America. And for this year, she became the president-elect of American Physiology Society, which means that for next years, she will be the #1 physiologist in America. Congratulations, Professor D, for your Alex. And we know Professor D for a long time. As you must know that Professor D ever came to our place in Palembang in 2010, Professor D, yeah? If you remember, yes. Yeah, in 2010, and she has gave the lecture to your seniors 11 years ago. Now, today, Professor D is still healthy, and yeah, we pray to the God that hopefully the Professor D is still always healthy to give the lecture to the world. And today, she willingness to give us a lecture to you after 11 years. Yeah. As you may know that Professor D is part of our family. Yeah, this picture I took this picture in 2011, Professor D, if you remember, in Rio de Janeiro. So we are family of physiology teachers, happy family. So for now on, that I also invite you after you finish your medical education, you can come to our place to become physiology teachers. Okay, and today Professor D will be give a very important lecture. I think the most important lecture about physiology. Professor D will tell us about the role of physiology on human body system and why we should learn physiology well to understand the medical knowledge. As you may know that student that we have a jargon that physiology today is medicine tomorrow. So which means that if you learn physiology seriously, so you will become a good doctor tomorrow. I think this is all for my introduction. And Professor D, you have we have arranged that you become a co-host, so you can share your presentation. And please take your time. And before that, please give applause for Professor D. Say hello to her. Okay, everyone, please unmute and say hello to her. Salam,
Hello,
ucapkan salam.
Professor D.
Professor.
Hello, Professor.
Okay, thank you. Okay, you may start, Professor. Yeah.
All right, let me see. Trying to see, I cannot see the chat. So if you will tell me if any questions are posted in the chat, and you can stop me at the time that you think is good to answer those questions. So I am delighted to be with so many of you today. It's evening it's Wednesday night in Texas right now. And I wish I was there to see all of your faces, because it is so much more fun for me and so much more interesting for me to be able to see you and see your reactions and hear what you have to say. So I hope that as we go, you will put questions and comments in the chat for me. And you can see here on this slide is a picture that I took in 2010 of the bridge there in Palembang. So first we have a poll, and I would like to know from you first, which organ system do you think is the most important one in the body? And the second poll question is, which organ system do you enjoy studying the most? So we'll just let you answer that. It should have popped up on your screen.
It's already open. You can share. You can click your answer now, student.
Okay, go ahead if you can get your vote in for us, please. And I will do what I do with my students when I poll, and I give you a countdown of 5, 4, 3, 2, 1. And now we will end the polling. All right. So share results. Can you see that now? I think. So most people thought the cardiovascular system was the most important by a big margin, and that's also the system you like to study the most. Okay. So let's go ahead and continue, and this will come up perhaps a little bit later. But as Dr. Irfan said, physiology is the center of many different fields in biology, as you can see. It overlaps with cell biology, neuroscience, biophysics, biochemistry. Proteomics and genomics is becoming much more important now in medicine, and yet it really reflects back to physiology. And then pharmacology, which I understand is one of your next modules that you will study. Anatomy, histology, and then of course, the one that is the reason you're here today is clinical medicine. Physiology is the basis for medicine. And when you're dealing with people who are ill, it is because something has gone wrong with their physiology. Physiology covers many of the different levels of organization in the body. And you can see here, it goes all the way from molecules up to populations of humans. And we're seeing more and more about population physiology, particularly with this COVID pandemic. But that's one of the things that I think makes it the most fun, is that it is not just focusing on one level. It has many different levels that you can think about. So I understand you're about to finish your first module in physiology, and I'm sure that it's very similar to the way students around the world study physiology. We have to study the body systems in a linear fashion because time is linear. So when your module began, and I don't know your syllabus, so this is probably not the right sequence of courses for you, but you start with something like cell physiology. And then maybe you look at the nervous system and the endocrine system and the immune system, then cardiovascular, respiratory or pulmonary, renal or urinary. And you notice we have two different names for these. Some of these are named for the anatomy, like cardiovascular, pulmonary, and renal, or we can call the same systems by their functional names of circulatory, respiratory, and urinary. And then finally ending up with digestive and reproductive, and now the module has ended. So we see the systems in a linear fashion as we're studying because time is linear. But physiology is not linear. Physiology is integrated, and that makes it sometimes difficult when you're finishing your first physiology course to see the big picture. And so since you are now finishing your first module, what I would like to do with you today is to tell you some things that we often speak about at the beginning of a course. But now that you have some physiology understanding, I think it will make more sense. And so I will pause at some spots, and I would hope, if you're a student, that when I say write down, that you will take advantage of that. You won't just sit there and wait for me to tell you the answer, that you will actually try to think, what do I remember? Because this is how you learn best, is to try and retrieve information from what you have already learned. So because physiology is integrated and not linear, the picture really should look something like this, with all of the different systems talking to each other and going back and forth in this very complex network. So today we're going to look at physiology as an integrated science, and my talk will be divided into three areas. For the first one, I'm going to talk about what we are calling core concepts. These are the big ideas of physiology, and they're the repeating patterns. And you can make your life much simpler as you go forward if you've learned the big patterns, because then you don't have to memorize 15 different little pieces. You say, oh, they belong into that pattern, and I know what that pattern does. So it simplifies what you're doing. And I think as you're beginning, sometimes it's hard to see patterns, but I'm hoping today, as we go back through these core concepts, you will go, oh yeah, I remember that. And now you start to see it as it appears in a different body system. The second thing we will do is I will take one of the core concepts, which is the most important for physiology, and that is homeostasis. And we will explore that in a little more depth. And then finally, we will talk briefly about pathophysiology, because I think as you finish your first course, this is where you're going now into your pharmacology course, and also following that into your pathophysiology studies. Because you've already seen most of the organ systems in your coursework at this point, I think it will help you to see these concepts now after you've studied the physiology. So core concepts are patterns that occur over and over. And there has been a lot of study done about how physicians think. And one of the things that we know is that when physicians make a diagnosis, they listen to a patient and they look at laboratory reports. They are actually doing at a subconscious level, not even aware of, finding patterns. They have learned patterns, and they see patterns in what the patient says and in the lab results. And so learning these patterns makes you better then at thinking your way through a patient when a new patient comes in. So there are 5 core concepts, and they're not unique to physiology. They are actually core concepts for all of biology. The first one is structure-function relationships, and there are many different aspects of that, and it's quite complex. We will be exploring all of these in a little more detail shortly. The second core concept, and it's also very broad, is pathways and transformation of energy and matter. The third core concept is information flow, information exchange, information usage, and information storage. And then our big one for physiology, of course, is homeostasis. And then number 5 is evolution. And we don't often in physiology talk about evolution, so I am leaving that out of my core concept discussion today. So let's look then at structure-function relationships. And what's fun about them is they go across all levels of organization. One of my favorites is the idea of compartmentation, the idea that everything is divided into compartments, usually with walls, but not always with walls. So for instance, the body itself is a compartment, and everything around us is our external compartment, and we have our internal compartment. And you can see that down here on the box diagram, which I suspect you've seen before, where this is the body separated from the environment by an epithelium of some sort. And then within the body, it's divided into 2 major fluid compartments, the extracellular fluid and the intracellular fluid. So everything coming and going to the cells must first go into the extracellular fluid after it has crossed that epithelial layer. So that, if you remember that pattern, it makes things much easier coming as you're thinking about where things are coming and going in the body. Then the other idea is that cell membranes, and this is a much more literal diagram. So you have over here, you've got cell membranes, and that makes each cell a compartment. And then here's your interstitial fluid. And then here is your endothelium, a layer of epithelium between the blood plasma and the interstitial fluid. And then within the plasma, you have the red blood cells and also all the white blood cells that are not shown here. So everything in this picture shows you the idea of compartments. And it becomes important later when we have to think about how do we get things around the body, whether it is material or it is some sort of information. Another structure-function relationship that's very important is the physical property of cells and tissues. And so shape and size are very important. This is a cross-section of the small intestine, and you can see the villi, and they're not blown up, but each one is lined with an enterocyte, and each of those enterocytes has multiple microvilli on top of it. And so in this case, the shape of the cells and the shape of the structure, the intestine, is actually designed to greatly increase the surface area. So surface area will influence transport, and so there is a structure-function relationship. Another physical property that's very important is the idea of cell-to-cell junctions. And this figure over here is showing you the structure of a tight junction with different fibers and such. And then we also have to think about extracellular matrix. And is it liquid like plasma, or is it something like hard like bone, or is it something in between like the basal lamina that underlies all sorts of epithelia? And then we think about, how do cells anchor to the matrix? So cell junctions are very important for function, because if you have a leaky epithelium, things are going to cross very easily. And that would be, for instance, that endothelium that we saw in the last slide. On the other hand, if you have a tight junction, the cells and the tissue can be much more selective about what is allowed across. Nothing, or maybe just water and sodium. So the function of the tissue then, it will depend to a large degree on the junctions. And then there are other mechanical properties that I know you've run across. So elastance and compliance and fluidity and stiffness and strength, all of these are properties that will influence how a tissue or an organ works. At the molecular level, structure-function relationships are also very interesting and important. And one of my favorites is protein binding. And so when proteins bind to other proteins or to other molecules, the protein binding has certain characteristics: specificity, it's selective in what it will bind; competition, where similar molecules that combine will compete for the binding site, and with differing affinities, then you may get different binding; and the idea of saturation. So here's a graph that I suspect you've already seen. It is looking at oxygen transport. This is the oxygen saturation here at a range of oxygen concentrations, PO2. And what you see then is, here's our standard adult curve, the red one, the maternal hemoglobin, but a slight change in the protein structure has significantly changed the binding affinity of the fetal hemoglobin for oxygen. And so the functions of the two hemoglobins are different, and their structure is different. This is another graph that I suspect you've seen when you studied the urinary system. And this graph is showing glucose and transport of glucose. So you've got the transport increasing with the plasma concentration, but then it saturates. And so you have reached your transport maximum. All of the available carriers are bound and working at full strength. One of the things that I think often gets ignored is that this is a universal property. Very often you see Michaelis-Menten kinetics when we talk about enzymes. But the reality is Michaelis-Menten kinetics apply to any protein. So they will apply to transporters on the membrane, like the one here. They will apply to the hemoglobin, and they will also apply to receptors, cell membrane receptors that are binding to some sort of ligand. So these binding properties, protein binding then is a universal trait for proteins that bind other proteins. The other molecular level relationship between structure and function that's important is solubility, and the idea of what is the solubility of a substance when it is in water versus the how lipophobic or lipophilic a molecule is in terms of being able to go through the phospholipid bilayer of the cell. So those are some of our molecular structure function relationships. Here's a picture that, again, you may have seen, and it's more oxygen transport on hemoglobin, again, in the adult. And we know that our total oxygen content in the arteries is a combination of how much is dissolved in the plasma, the PO2 of the plasma, and we know that's relatively small, plus the oxygen bound to hemoglobin. And there's your protein binding again. So on the left side here, what we're looking at are factors that are going to influence how much dissolves. And you see that many of them are physical traits. So airway resistance, lung compliance, that's telling you what's the tissue in the lung is like in terms of being able to stretch and be elastic. The surface area, the membrane thickness, is there scarring, or there depositions, or is there pulmonary edema that is influencing the oxygen diffusion? So these are all physical properties of that exchange surface in the lungs, as well as the exchange of gas between the atmosphere and the alveoli. On the other side, it's protein binding again. So we can see here's saturation. Is it possible to occupy all the binding sites? And then here are different factors that might influence it, particularly pH and temperature. So when you're aware of these things and you see them, it makes the commonality, the ability of proteins to have saturation, specificity becomes important. Here's our second of the big ideas, and this is pathways and transformation of energy and matter. So we'll start with energy, and we are particularly interested in energy stored in chemical bonds. And here's ATP being used to facilitate a chemical reaction. So here's our overlap then with biochemistry. And then the energy that's stored in biomolecules, like the lipids that are stored in adipose tissue. So we have potential energy stored in chemical bonds. And we also have potential energy stored in gradients. And so we can have concentration gradients storing energy. We can have electrical gradients storing energy, or as this sort of representation of a typical cell shows, we have electrochemical gradients that combine the electrical gradient and the concentration gradient. Transport is another pathway or idea that falls under pathways of energy and matter, because now we're talking about pathways of matter. How do substances get from one place to the other? And there are three general forms of transport. One that I know you've seen is the bulk flow of fluids. And engineers consider both air and blood to be fluids, interestingly enough. The air is a compressible fluid, and then water is a non-compressible fluid. So water is liquid and air is a gas, but they are both fluids and they obey the principles, the physical principles of fluid flow. So if you have learned the rules about what governs blood flow, like resistance, radius, pressure gradients, etc., in the cardiovascular system, then you can apply the same principles when you study the pulmonary system. Here's diffusion. This is very simple concentration gradients, and it can either be not across a barrier. So this figure is showing you autocrine and paracrine signals that are released from a cell into the ECF and simply move by diffusion to act on neighboring cells. It can also be simple diffusion through a phospholipid bilayer, through a cell membrane. And then we also have membrane transport. Protein-mediated membrane transport, such as this is showing a channel, but we also have carriers that don't ever form an open pore. And we also have vesicular transport, endocytosis and exocytosis, where the only way to get large substances across the membrane is to have a vesicle that fuses with the cell membrane, opens up, and then its contents can move out by diffusion. Next important concept, mass balance. And here's our schematic figure where the load here, and so this is looking, for instance, this applies to the body load of any substance. It's often used for sodium, for example. But to maintain a constant body load, anything that comes in must leave. And it can leave either by excretion or, depending upon what it is, it could potentially be metabolized to a different substance. But related to mass balance, and people often don't think about this, is clearance. And if you just studied clearance in the kidneys, guess what? It's not just a kidney function. Excuse me. What you're going to find when you start into pharmacology is that clearance is used for the clearance of drugs from the body. And that can be in the urine, but it can also be by the liver. And it can also be by cells taking up the substance and metabolizing it to something else. So clearance is a universal property, not just for the kidneys. And then you may have studied the Fick equation that can be used for calculating cardiac output. And here's an example. This is simply mass balance, where you've got arterial oxygen coming in and the cell is taking some out, and then what's left over is going off on the venous side of the transportation. And so you can then use this concept of mass balance to create the Fick equation. Our third concept, core concept, is information flow, exchange, and storage. And what everybody immediately thinks about with this is DNA, RNA, and proteins. And then the processes of transcription and translation by which the code in DNA is transcribed to RNA, and the RNA then goes and is translated into a string of peptides. But there are other ways that we can exchange information. One of them is the idea of cell signaling. How do we get information across that compartment wall that is the cell membrane? And there are two ways, and this goes back to our principle of solubility. If it is a lipid-soluble signal, it can just move into the cell and find either a cytoplasmic or a nuclear receptor. On the other hand, if it is a lipophobic signal molecule, it cannot get into the cell. And so it has to pass its message from the extracellular space into the intracellular fluid. And it will do this with a receptor on the cell membrane and some form of signal transduction. So that is also information exchange, moving information from one compartment to the next. And a lot of our information is used then for cell-to-cell communication, which is critical for coordinating the events in a large multicellular complex organism like the human body. And in this case, the signals, as we saw earlier for gradients, can be either chemical signals or electrical signals or some combination thereof. So cell-to-cell communication can take place at the local level, and you see that here, where it's only around the blood vessel. At the simplest, it can be through gap junctions. We think about gap junctions in the heart, in the intercalated disc, electrical signals for smooth muscle that are coordinating all the muscles to contract together. We have electrical gap junctions in the brain where we're getting electrical signals passing. But we now know that almost every cell type in the body can have gap junctions and use them to pass information to neighboring cells. So there's no need to release the signal into the extracellular fluid. Then local signals, we already saw the idea of paracrines and cytokines that are released from one cell and act on a neighboring cell. And then there is contact-dependent signaling. And this is unique pretty much to the immune system, where an immune cell will present a piece of antigen on its surface, and then another immune cell comes over and must contact that antigen in order to initiate a process. And then the diffusion, the paracrine and autocrine signals, as well as the ones we call cytokines. So those are your different types of local cell-to-cell communication. Then we have long-distance communication, and that is particularly the hormones and in larger concentration cytokines that get into the bloodstream and are carried throughout the body. And then we have the nervous system, which uses a combination of chemical and electrical signals. So the rapid electrical signal of the action potential going down to an axon terminal that then releases a neurotransmitter that diffuses across that tiny gap we call a synapse to the postsynaptic cell. So now I've got another task for you. Let's see if you can take what I've just been talking about, core concepts, and recognize them when you learn some new information. So I chose something that is of great interest to everybody in the last year, which is the mechanism of action of the coronavirus SARS-CoV-2 and what it does at its target cells. And so what we see here is the virus with its spiky proteins. And the viral spike protein then comes in on the target cell and binds to the surface enzyme called ACE2. And then there's a neighboring enzyme, which is a serine protease, and it actually then activates the ACE2 and cleaves the spike protein. And after it's done so, this combination of events causes endocytosis of the viral particle into the cell. And once it gets in, the RNA is released and it goes over to cause the production of new viral proteins and new viral RNA, because this is an RNA virus, which are then packaged, and then the new replicate viral protein particles are being released from the cell. And we see here an antibody binding to the spike protein and preventing it from going and binding to ACE2. So how many different core concepts have I illustrated in this process? So think about this for a second, and then we're going to have another poll question. So look through this and see how many of the core concepts did you see? And the poll is open now. All right. There we go. Now we get some people voting. All right, I'm going to give you a countdown. So 5, 4, 3, 2, 1. Boy, a bunch of them are coming in all at once. Okay, let's end the polling and see. Oh, let's try this again. Okay, you should be able to see it now. So most people said 3 core concepts. All right, so I will show you, there are a couple of different ways you could possibly break it out. So there was our question. And the question is, how many core concepts and what are they that you see here? Well, one of them is protein binding. And so there was protein binding in 2 different places, the antibodies binding to the spike protein, as well as the spike protein binding to ACE2 and also to the serine protease. So that's number 1. The next one that's obvious is the idea of membrane transport with endocytosis of the particle and then exocytosis of the replicated virus particles. So those 2 probably were easy, and you might have made 4 out of them instead of just 2. So we don't care exactly what the number is as long as you recognized each of them. Then we also have the idea of cell signaling, because it is not until you get this process of binding and then the spike protein being cleaved that it will trigger their steps in between that we did not talk about, but that triggers the endocytosis. So that is a form of signaling. And then, of course, and the words are right there, information storage and transfer with RNA both being transcribed and then translated to make new proteins. So if you have an awareness of this, it helps you sometimes to break down new information when you encounter it for the first time. So now we come to our last and our biggest and most important concept for physiology, which is homeostasis. And so essentially we try to maintain homeostasis, but we can either have external influences or sometimes internal influences that result in the loss of homeostasis. And the body will attempt to compensate, but it doesn't always have the appropriate mechanism available. And so if compensation succeeds, then we maintain a state of wellness. But if it fails, then we go into a state of either illness or disease. So homeostasis then for us is really trying to maintain a relatively constant internal environment to keep the body functioning. And you saw this, something similar to this before. This is another way to show that here are the cells in the center, and everything outside has to go through the extracellular fluid. So the extracellular fluid then is what the body monitors and tries to use to maintain homeostasis. So all of our homeostasis really is aimed at keeping the brain functioning. And when people were talking about the most important system of the body, I mean, I think in many ways you would have to say the central nervous system, certainly, because if the brain goes, it doesn't matter if everything else is functioning perfectly. And so homeostasis of the ECF is really aimed at keeping the brain functioning properly. And so this is a way, this is sort of a control system diagram, and we will have a regulated variable of some sort. And as long as it stays within our homeostatic range, our desired range, nothing needs to happen. But if it moves outside of the desired range, there is a sensor that is activated. And so I think many times one of the ways to think about the regulated variable is to say, what is the variable that is being monitored? And that's important because that is the regulated variable. So the sensor is activated, sends a signal to a control system, which is usually an endocrine gland or cell or the nervous system, which then creates some sort of output that goes to tissues that are the targets or the effectors. And those effectors then will influence the regulated variables. So we're looking at our typical reflex, homeostatic reflex pathway. So I have another task for you, and I hope you will do this. We don't have a way to poll for it, but I would like you to take a minute or so and list as many regulated or monitored variables in the ECF as you can that our bodies use to maintain homeostasis. So I'm going to turn my music back on, and we'll give you about a minute or so to work on this. All right, that's getting to be about enough time, I think. You should have been able to get things that way. All right. So let's see how many of them you got. Oh, hush. There. Okay, let's look at my list, and you can check your list against what I have. And one of the things to remember when you're doing this is to try and be as specific as possible. So Blood pressure and blood volume, which are related but not the same. That's both of those are monitored variables. Oxygen and carbon dioxide concentrations in the blood. Glucose concentrations in the blood. Why is that important? Well, oxygen and glucose are essential for the brain to function. Except in times of starvation, the brain cannot use fuel other than glucose for its energy substrate. So this is one of the reasons we tightly regulate our blood glucose concentrations. pH is another one. So hydrogen ion concentration, osmolarity, and again, you might not think of osmolarity as being important for the brain, but if your osmolarity goes too low, what happens? The cells swell, and when the cells swell, where are they? They're inside a bony case called the skull, and so now you're going to have pressure on the brain, and so having swelling because the osmolarity is too low can cause the brain to fail. And then two ions specifically, potassium, and you should be able to tell me why potassium is important, because it is the primary ion responsible for resting membrane potential. So if potassium goes out of range, you're going to set off all sorts of chains of events due to hyperexcitable neurons or hypoexcitable neurons. And the same thing with calcium. Calcium is regulated tightly because through a rather complex pathway, changes in calcium actually change sodium permeability in the cells. And so that then will affect the neural excitability. You notice that I haven't said sodium. We do not monitor sodium per se. All the sodium regulation is done because we are monitoring blood pressure, blood volume, or osmolarity. And then core body temperature is our last one. So there's your list of all the variables that, or all that I can think of certainly, that we monitor in the extracellular fluid. All right, so in order to maintain homeostasis, we have to have communication, and there are three primary control systems: the central nervous system, the endocrine cells, the endocrine glands and cells, and immune cells. And they must work with the other organ systems to achieve a common goal, which is to maintain homeostasis. And so here we see the interrelationship. Immune cells put out cytokines. The brain puts out neuropeptides primarily and other neurotransmitters. Endocrine cells put out hormones in different flavors, if you will, steroid, peptide, and amino acid derived. And they're all working together on each other as well as on the target cells. And so when something happens, either pathogens that activate the immune cells or some sort of physical or emotional stressor, and we're using that in a very broad sense, then all of these systems will come into play. So the organ systems also have integrated function. We talk, anatomists talk about the lymphatic system, and what I teach my students is that's anatomy, but because the anatomical lymphatic system actually functions in three different physiological systems. In the digestive system, you see the green lacteals here that are used to absorb and transport absorbed fats back to the bloodstream. And then in the circulatory system, we have our lymph capillaries that are picking up excess filtered fluid out of the blood capillaries and returning it to the circulatory system. And then, of course, our traditional immune function, and this is showing a lymph node with the immune cells. But all of this fluid from the lacteals, as well as from the systemic capillaries, is being picked up and then filtered through these lymph nodes on their way back to being put into the circulation. You've probably already seen the overlap in the control of blood pressure. So if we think about the factors that influence mean arterial blood pressure, we have cardiac output, and in this case, blood pressure is the monitored variable. You know, you have baroreceptors for that, but it is controlled by changing heart rate and stroke volume. We do not have any mechanisms for directly monitoring heart rate or stroke volume. We simply monitor the pressure and change the two factors to get the pressure up. Then we have the resistance of the system to blood flow, as well as the relative distribution between the arterial and the venous sides of the circulation. So here's mean blood pressure and cardiovascular control mechanisms. But there's a fourth one, and it's not part of the cardiovascular system. It's actually blood volume. And blood volume then is going to be a matter of mass balance. What's our volume? It's fluid intake and fluid loss. And so fluid loss then can be passive, either blood loss, excessive sweating, and then it can also be regulated by the kidneys in times when there is a need to increase fluid loss because the blood volume has gone up. So here we see already now that blood pressure involves the integration of two different systems. Here's another figure showing the same thing, which is a decrease in blood volume will be reflected in a decrease in blood pressure. And then you've got both volume receptors, which occur in the atria, as well as carotid aortic baroreceptors. And so now you've got, if low blood volume, you get a cardiovascular response, you get a renal response, and you also get a behavioral response, which we don't too often think about when we're talking about physiology. But thirst is a very powerful drive that will send you to go get fluid if you are low on fluid. And then here's the opposite, high blood pressure. And again, here it's a cardiovascular and a kidney response because we don't have any way to get rid of volume voluntarily. pH balance is another one that is integrated between more than one system. We've got, here's your homeostatic range for pH, and we have our first line of defense as our buffers. But then after that, we have to rely on the ventilation, on the pulmonary system to either retain or get rid of CO2. And then the kidneys, which can, if it's a matter of output, get rid of hydrogen and reabsorb bicarbonate to add to our buffer, or get rid of buffer and absorb hydrogen according to what the need is. So here's another figure showing how complex this integration can get. This is showing the response to dehydration with a hit up here at the top, decreased blood pressure, decreased blood volume, and increased osmolarity. So this is someone who's dehydrated. And each of these colored vertical bars represents a different system working to try and maintain homeostasis. So here is the blood pressure response that we've been talking about, going down with cardiac output and vasoconstriction. Here's our renin-aldosterone or renin-angiotensin system, which has become much more interesting now that we now know about the way COVID virus gets into cells. But here's angiotensin 2, and it turns out angiotensin 2 goes everywhere and integrates all of these different systems, because not only does it work at the level of the adrenal cortex, but it also acts directly on arterioles for vasoconstriction. It goes up and acts on our medullary cardiovascular centers to help raise blood pressure. It goes over to the posterior, the hypothalamus, and tells it to release vasopressin from the posterior pituitary. And it acts on the thirst centers, sending the person, if they are capable, to go find more fluid. So here is a case of one signal molecule that is integrating all these different systems to try and achieve a common output. And then here's another one that is one of our popular ones, which is the immune cells. The immune system overlaps and integrates with all of the other systems practically. So obviously the integument, the skin, is a critical place. But then we've also got the other systems that have exposure to the external environment, our respiratory system, digestive, reproductive, and urinary systems. And so those systems then are going to be part of the defense mechanism carried out by the immune system. And they're going to use barriers of their own, such as mucus, but they're also going to rely on secreted antibodies and structures such as cilia to try and flush the pathogens away. And then if something makes it past the barrier, then all of these different systems have what is called mucosa-associated lymphoid tissue, or MALT. And these then are the immune cells that are associating with all of the different other organ systems to try and protect the body from invaders. So let's, for our last little bit, let's talk a little bit about pathophysiology and what happens, because I think it's when you get into the pathologies that you see the integration of system function the most. And this is where trying to understand mechanisms, what the normal physiological mechanism is, becomes critical in terms of treatment as well as developing new treatments. And before this year, the example I would use when I teach is diabetes, because both type 1 and type 2 diabetes will cause a series of integrated responses that go across multiple systems. And fortunately, we have a pretty good idea for most of them what the mechanism is, so it has been possible to target good treatments. But in the last year, we have been dealing with COVID-19 and this coronavirus, whose mechanisms are not really known. And so I will we'll talk a little bit about each of those. So this is another one of those, you can't read it very well because you're not supposed to, but this is a map of what happens to a type 1 diabetic in diabetic ketoacidosis, DKA. And it's showing the metabolic changes that happen in the absence of insulin, and then all of the metabolism that's leading to the hyperglycemia. And if it's bad enough in terms of not getting things, nutrients into the cells, then we go into ketoacidosis. And now we have a metabolic acidosis. So go back to that picture of pH balance. That's going to involve the respiratory and the renal systems. And then we're going to have dehydration because of an osmotic diuresis. So now that goes back to that map of dehydration with the increased osmolarity and decreased volume and pressure. So here's a case where the pathology is triggering responses in multiple systems. It's the dramatic one. It is the least common. But even in type 2 diabetes, we see the overlap of systems. So we have problems in the eyes that are actually part of the blood vessels in the eyes, diabetic neuropathy, which we still don't completely understand, chronic kidney disease, peripheral vascular disease, particularly in the microcirculation, that leads to gangrene and amputation of limbs, and then gastroparesis. So this is a case where the one disease is having multiple functions, and when one system goes down, it may take others with it. COVID then also is a good way of looking at a case where we have integrated function and we don't really know yet completely what's happening. So for instance, we can see these are the tissues where ACE is expressed, and obviously the upper airways and the vasculature in the lungs and the gut are the primary ones in terms of where the virus enters the body. And then we've been hearing a lot about the heart because of all of the cardiovascular complications that seem to be occurring in certain people, as well as the microclots and what's going on in the vasculature. So here is a complex disease that we don't really understand. But we can look at it in terms of the physiology that we do teach. So before this year, I never taught about ACE2 and never really thought about it in terms of where it came in the normal RAS pathway, renin, angiotensin, aldosterone. And so here's our angiotensin 2 that we saw in the previous slide, the map of the dehydration acting on an AT1 receptor. And that's our classic arm where it's raising blood pressure, vasoconstriction, release of aldosterone, and also is pro-inflammatory. And I see somebody has a hand up, and I will take that at the end of this series of slides. And then here's where ACE2 comes in. It breaks down angiotensin 2 to angiotensin 1 to 7. And angiotensin 1 to 7, it turns out, actually has its own receptor called MAS, and it is the counteracting arm. It is protective. It decreases blood pressure, causes vasodilation, and is anti-inflammatory. So what we're looking at is another case of homeostasis. In the normal person, these two arms of the pathway are balanced so that you've got ACE on one hand raising blood pressure, and then you've got ACE2 helping decrease it. But what happens when the system now goes out of balance, either by drugs such as your antihypertensives that act on receptors like the angiotensin 1 receptor or the ACE inhibitors? And then SARS-CoV-2, of course, now is binding to ACE and causing it to be removed from the surface of the cells. So here's our picture. Angiotensinogen converted by renin to angiotensin 1 to angiotensin 2 to the AT1 receptor raises blood pressure. But what we see is ACE2 now is part of the regulatory, counter-regulatory pathway, and it takes angiotensin 1 to angiotensin 1 to 9, as well as 1 to 7. And both of these then are counter-regulatory. But what happens then if you give somebody an ACE inhibitor, and what happens if you give them an angiotensin receptor blocker, which we call an ARB? So see if you can predict based on the mechanisms what's going to happen to ACE2 concentrations in someone who is taking one of these drugs. So let's walk through it. If you give someone an ACE inhibitor here, what's going to happen to the concentration of angiotensin 1? It's going to go up. And so increasing substrate chronically is probably going to cause the cell to upregulate its production of ACE2. And the same thing with ARBs, where you're blocking here, and so the spillover is going to go through into this pathway. So you would expect then that the cells are going to upregulate ACE2 in response to increased substrate. Well, does that mean then if they're upregulating ACE2, and ACE2 is how the virus gets into cells, the coronavirus gets into cells, does that mean people with more ACE2 are going to have worse disease? And so by understanding the mechanism, it generates a testable hypothesis. And so the prediction might be people who take ACE inhibitors and angiotensin receptor blockers have more ACE2 and therefore are more likely to develop COVID-19 or to have more severe disease because they will have a higher viral load in the body. And indeed, last year, about a year ago, there was an article in the British medical journal, The Lancet, that predicted that people treated with ACE inhibitors and ARBs for hypertension would have more severe COVID-19. Well, let's see what happened. The evidence turns out that that's not what happens. By the summer, observational studies suggested that the drugs were not harmful and could even, for some reason, be protective. And then a review of all sorts of studies that were being done throughout the year that was released in November 2020 said taking an ACE or taking an ARB does not increase the risk of COVID-19, and it is not associated with more severe disease. So here's a case where we based a prediction on a mechanism that we knew about, but then in reality, it was much more complicated than that. And that is because of the integration of physiological mechanisms. We still don't know why it is that they are not at increased risk. So let's see. I'm going to go ahead and look at the chat and see if there's anything there for me to answer. Okay. Irfan, did the person that raised their hand have a question that you know of?
Yes, I believe so.
Okay.
Rob, please, siapa yang tanya tadi? Silakan langsung jawab. Silakan langsung tanya. Ya. Raise hand. I see someone raise hand just before. No? Not anymore?
Not anymore.
Yes, maybe later, Professor D. You can continue now.
Okay. Well, this is, I think we're pretty much going to stop here because I think I'd like to see if people have questions.
Okay.
And so, okay. Yeah, go ahead.
Actually, we have one question from Dr. Adrianta. I special invite him to join the Zoom.
Oh,
Dr. Adrianta, please give your question. Halo? Udah masuk belum Dr. Adrianta nyata? So let me read his question. Please ask Professor D. What do you mean that the blood pressure is more important compared to cardiac output to regulate cardiovascular system?
Yeah, it is not more important. It is the monitored variable.
Oh.
So there's a difference, and we do not make a very good distinction with our students, but there's a difference between a regulated variable and a controlled variable. So blood pressure is the regulated variable. That is the one we have the sensor for. We have carotid and aortic baroreceptors. They're monitoring pressure. But they get the pressure back in range by controlling other factors. And so they are controlling heart rate, force of contraction, and vasoconstriction. So those are controlled variables that are used to regulate blood pressure. But blood pressure is the monitored variable. It is the one for which we have a sensor.
Okay.
Just like and the other example of that is osmolarity. We do not have sodium sensors. We have osmoreceptors.
Okay.
But when an osmoreceptor is triggered, it is going to have effects, depending upon which way, potentially on vasopressin and water, as well as aldosterone or possibly the atrial natriuretic peptides.
Okay.
So those are the controlled variables. The monitored variable is osmolarity. Does that, the monitored or regulated variable is osmolarity.
Okay. And the second one is like brain has their own regulation to control themselves about the blood pressure. How about the coronary artery in heart? Is there any local auto-regulation for them?
I believe there is a little bit of, I think all of the not the arteries per se, but I believe the arterioles throughout the body, including the heart, have significant amounts of local regulation. So adenosine, oxygen, CO2, pH, all of those factors can work at a local level.
Okay.
And that's where nitric oxide comes in.
Yes, of course. Yeah.
And the pills that people take, the nitroglycerin pills that increase nitric oxide.
So, student, do you have any question? That I believe this your lecture is answer for all of the professor. They already done the tutorial session, problem-based session, for 3 times, if I'm not mistaken. And this is your lecture is the answer all of this problem. Okay, student, silakan kalau ada yang mau tanya tentang skenario kemarin yang masih belum paham, ayo, inilah kesempatannya. Mumpung bertemu langsung dengan orang yang mengarang bukunya, ayo silakan. Ayo beraniin aja.
Dr. Irfan.
Oh, Dr. Adrianta. Okay, please.
Thank you, Prof. D. How are you? Nice to hear your lecture.
I'm going to come back to, let's see, stop share. So maybe I can see you.
Okay.
There we go.
Yes.
Okay.
I'm at home. Yes. Nice to have your lectures. And I was always difficult for the students to explain the regulation of the cardiovascular system, because you mentioned you have to stress now the variables that you are controlling and the other that is not controlled. Like cardiac output is not a variable that is monitored, isn't it? It's the blood pressure that you have. And also when we talk about long-term, we talk about renal and we talk about hormones, but that is also not a variable that is regulating. It's the osmolarity or volume, sorry. It's the volume that is important. So I now have to switch my lectures to stress more about this, not about the cardiac output. We know cardiac output depends on heart rate and stroke volume, but that is not monitored by the system, isn't it? Can you explain more so that we all know for the students? Thank you.
So I use for this, and I did not put it in my slides, but this is where I think it's very important to use reflex maps. And so you would have the stimulus is low blood pressure. And then the sensor for that is carotid aortic baroreceptors.
Yes.
And then you have sensory neurons into the brain, into the medulla and the cardiovascular centers in the brain. And then it has to, and I'm saying in quotes, decide, it is programmed to take the input and make an appropriate output.
Yes.
So it will say blood pressure is low everywhere. Therefore, I need to do the following things to raise it. I need to increase cardiac output. I maybe need to constrict veins and put more volume to the arterial side. I need to vasoconstrict the arterioles so that there's less outflow from the arteries and my arterial pressure stays up. So it's really what is being monitored is arterial pressure because, and if you think about it, it's pressure to the brain.
Yes.
Again, because it's all aimed at maintaining blood flow to the brain.
No.
So
Yeah.
a good example, I think, to think about, do you know the mammalian diving reflex?
Yes.
Where you can have a student put their face in water and their heart rate for most of them will decrease. But if you are checking it, you're getting different output than you do when you have low blood pressure. The pressure was vasoconstrict at the periphery, because if you are a whale or a seal, you're in cold water, and so we don't want to lose heat. So we vasoconstrict, which sends blood to the core, which raises central pressure. So now we need to bring the pressure down. So we have vasoconstriction combined with decreased cardiac output. So that's a case where the brain is able to integrate the different input and realize I'm not trying to raise blood pressure. I'm trying to counteract a different mechanism, the vasoconstriction because of cold. It's complicated. Does that make sense though?
Yeah.
Thank you, Dr. Adri. So Professor D, we have one young girl to raise her hand. Anandita, please give your question to Professor D.
Okay, Dr. Irfan, thank you so much. Hello, Professor D. I want to ask you a question. It's actually about our case. We have a patient with hypothalamic shock, and the thing that we actually confuse is this patient also show symptoms about the diaphoresis. He's having diaphoresis, and diaphoresis is a abnormal sweating, right? And the thing that we confuse is this patient losing a lot of fluid, but the body is excreting the fluid itself. So I think how could that happen, Professor? Thank you so much.
You're welcome. It's a good question. So not everything that happens is good. I mean, you have an example right there where one of the programmed responses that the body is programmed in, if this, then do this. And it happens that is not a good response. That is, as you say, it's going to make things worse. So, and that is just a matter of programming. And with experience, you learn where they are. But ideally, the diaphoresis will not be severe enough to make the fluid loss as it would if you were out working for hours in a hot environment. So it's a little fluid loss, but it's part of the overall mechanism. So sometimes things are not always adaptive. They're not good. And sometimes we don't have a mechanism to cope with a particular change. So you will learn these as you go along.
Thank you so much, Professor.
Okay.
Thank you for your
Okay.
question.
Okay. Is there any question for anyone? One question. Okay. If not, Professor D, you can continue or not? Yeah.
No, that was my last slide.
Okay. Okay.
So all I had at the end was to say thank you to you all for your attention and for being here.
Okay. Do you have any question from faculty? Prof. Hardy. Hello, Prof. Hardy. How are you?
I'm fine.
Okay.
Thank you very much, Dr. Professor Didi. Nice to meet you.
So nice to see you.
Mana Prof. Hardinya? Nah, that's
Hi.
right.
Long time to see you.
Yes, I know.
Yeah.
Very excellent lecture.
Thank you.
Thank you. Thank you
Adik-adik
for
ada yang ingin
coming.
ditanyakan enggak?
Nggak ada.
Hah?
No questions.
Oke. Yang mahasiswa, ayo. Mestinya banyak tuh yang ditanyain dari skenario kemarin. Come on. Dari itu. Please, we wait for a while. Mahasiswa, ayo. No question?
No question. So remember all of these core concepts. Start pharmacology, and as you start your pathophysiology and clinical blocks, because those same core concepts, particularly protein binding and clearance, those are all going to be everywhere in pharmacology. Very important.
Okay, we have question from message to me, direct message to me from the faculty member from other university, Professor D. That you mentioned that the factors that is important for brain function is potassium concentration. So how does the body monitor specifically potassium concentration, since the body does not have receptor that monitor it? For sodium, okay.
Yeah, but potassium we do. The adrenal cortex, the cells that secrete aldosterone, are directly sensitive to potassium.
Okay. And for the sodium concentration, the body might sense changes indirectly through change in osmolarity or body fluid. But what about potassium, Service Minda? Is there any influence of potassium for the body fluid?
On the body fluids, no, because potassium is, well, as if you are putting out aldosterone, if you're putting out a lot of aldosterone, you have the potential to excrete too much potassium.
Okay,
so I mean, yeah, and that's one where, you know, correcting one problem creates a new problem.
Okay, yeah. Correcting one problem, create new problem. Yeah, good.
Yeah.
Very important.
Good quotes.
Yeah. So, student. If not, that we have to end the session because Professor D maybe already sleepy now. 9:30 p.m. in Austin now. So, but this is very, very, very rare opportunity for you. Yeah.
Yes,
So,
I wanted to. I would rather be there with you, though.
Yes.
Yeah,
Of course.
much more fun.
Yes.
Maybe next year.
Okay. So I believe that we will invite you again, Professor D, for another lecture for next the future session. So I think if there is no any question from you, so maybe I think this is all our session today. Maybe before we end the session, maybe Prof. Hardy want to say something that for Professor D.
Thank you very much, Professor D, for your excellent lectures. You have been, since our last meeting, maybe 10 years ago in Palembang,
Yes.
the International Conference of Physiology.
Yes.
It impressed me so much. Our Department of Physiology here.
Yeah.
Now I'm busy in the COVID-19 task force.
Oh, yes, I'm sure.
Yeah. Many questions about the COVID. The one you mentioned about the ACE inhibitor, using the ACE
Yeah.
inhibitor. Thank you very much.
Yes.
Hope to see you soon.
Yes, I hope so. Thank you very much for having me to do this.
Welcome.
Okay. Okay, I think Prof. Hardy, thank you very much for your nice closing speech. And to Professor D. Silverthorn, so on behalf of our Dean, actually I am the Vice Dean now, and on behalf of our Dean, we would like to say thank you for you, and then we hope that you are willingness to give another lecture in the future session. So thank you, and student, please give a big applause for Professor D and say thank you to her. Oke, silakan adik-adik semua.
Professor.
Thank
you so much, Professor.
Professor D.
Thank you.
Okay. And we hope that we can see you in next year in Beijing and Tianjin.
Yes.
I asked for all the faculty members to save money now, so we can go together to Beijing next year.
Semoga next year
Yeah.
udah nggak corona
Okay.
lagi.
We hope that. Yeah. And like Professor D said that after Beijing in 2025, we will go to Germany, southern Germany or Austria, to see the Liechtenstein Castle. Okay ya, sampai ketemu lagi. Thank you very much, Professor D. I think that's all for today, and I hope that you are still healthy, and congratulations for your new position as the president-elect of the American Physiology Society. So we hope that we can see you again for the next moment. Thank you. Bye-bye.
Thank
Terima
you.
kasih.
Bye-bye.
Ya, terima kasih.
Bye-bye.
Ya,
Thank you, Prof. Thank
Bye.
you.
terima kasih. Ya.
Thank you for coming.
Oke.
All right.
Bye.
Bye.
Bye,
See you
Professor.
when I see you.
Thank you.
Dia tuh ketemu anak aku. Iya, iya. Adik-adik, habis ini kita kuliah saya ya. Videonya sudah dilihat belum? YouTube-nya. Nah, saya kasih kesempatan untuk nonton YouTube. Kuliah saya kayaknya topiknya mirip-mirip dengan Profesor D tadi, tapi tidak selengkap Profesor D. Nanti saya hanya konsep-konsepnya aja ya. Jadi lebih ke pemahaman sebagai pesan terakhir kami lah ya, sebelum Anda nanti berlanjut ke blok-blok berikutnya. Karena habis ini nanti kalian akan belajar pada blok-blok sistem ya. Jadi saya akan memberikan resume ya. Jadi mungkin lebih sesuatu yang disampaikan Prof D tadi kita akan sampaikan lagi dengan cara yang lebih simpel ya. Oleh karena itu silakan ikuti kuliah YouTube-nya dulu ya. Link YouTube sudah dibagikan, sudah ada di link YouTube. Tonton dulu YouTube-nya, nanti mungkin dalam satu jam ya, 10.30 nanti ketemu saya lagi pada kuliah final dari blok sistem tubuh ini ya. Oke, sampai ketemu.
Sampai ketemu, Dok. Terima kasih banyak, Dok.
Terima kasih banyak, Dok. Terima kasih banyak, Dok.
Terima kasih banyak, Dok.
Terima kasih, Dok.
Terima kasih, Dok.